Welcome to the fascinating world of neurons, where tiny protein complexes work tirelessly to keep our brains buzzing with activity. Imagine a bustling city where every delivery truck needs to drop off its cargo smoothly. That's what synaptic vesicles do at the microscopic level. They fuse with the presynaptic plasma membrane, releasing neurotransmitters that carry messages across the synaptic gap. These neurotransmitters are like little postal carriers delivering signals that keep our thoughts sharp and moods balanced. These processes rely on the intricate cast of synaptic proteins, including synaptosomal-associated protein 25, or SNAP25, as well as syntaxin and synaptobrevin. These are molecular matchmakers, forming a complex that acts like a zipper, bringing vesicles and membranes together to facilitate fusion. These proteins are often hydrophobic or hydrophilic, meaning some left to hide in the oily inner parts of membranes, while others prefer the aqueous environments outside. It's a delicate dance of molecular compatibility. What happens when this fine-tuned system goes astray? Enter neurodegeneration, where things get messy. One culprit is amyloidogenesis, the formation of amyloid plaques, stubborn insoluble ligaments that tend to clump together, especially in disease like Alzheimer's. Some of these proteins are helical, forming cell-like structures that can misfold and stick together, creating tetramergic aggregates. These abnormal assemblies can cause complex dysfunction, especially when they interfere with neural signaling. Within the brain, regions like the orbital frontal cortex are critical for decision-making and emotional regulation. When synaptic proteins or their isoforms, different versions of the same protein, are disrupted, neural communication falters. This can lead to issues like presynaptic overstimulation, where neurons fire too much, or problems with the actin cytoskeleton, the structural framework inside cells responsible for cell motility, neuronal differentiation, and maintaining intercellular connections. These processes are vital during development, guiding cell migration and ensuring proper tissue formation during mitosis. Neuronal transmembrane proteins such as neurexin serve as molecular bridges between neurons and glia, supporting myelination of axons and the formation of dendritic spines, the tiny protrusions where most synapses occur. The axon itself, covered by the axonema, acts as the highway for electrical signals traveling from the center or cell body to the axon terminus. On the surface, receptors like AMPA receptor subunits respond to neurotransmitters modulating neuronal activity. Inside the cell, lipid metabolism and fatty acid binding proteins maintain membrane integrity and energy supply, ensuring that all of these processes run smoothly. So next time you marvel a clever thought or a beautiful sunset, remember the tiny bustling world inside the brain, where proteins, vesicles, and signaling pathways dance together in a delicate yet resilient symphony of life. Just like that, science continues to uncover the secrets behind the most extraordinary organ, one molecule at a time.